Welcome to my analysis of the Big Y test, which is offered by Family Tree DNA. And the best place in my experience to get the analysis is YFULL.com. This is the fourth talk on Y-DNA, and this will be the final talk in this series. The first thing to understand is Y-DNA, especially the Big Y, is still a little pricier than other things. But a look here at the review of the past costs. The first DNA tests were taken back before 2002, and in the year 2000, that big test cost $3 billion. In 2018, now, it costs less than $1,000 to do that same test. And it's estimated that in the next 5 years, that cost will drop below $100. So these tests that we're talking about may seem pricey to you now, but if you start collecting the DNA and saving it at a place like Family Tree DNA, where you can test later, they keep your samples for 20 years at Family Tree DNA. So if you collect the people you think you might want to do right now and save them until the sales come up or until the prices get to a place you like, then you can do the Big Y test. So let's move forward and understand what that test might give you. The goal of the Big Y was to read virtually all of the Y chromosome that was useful for genealogical purposes. So what works for genealogy? Not just all of it, not the medical part, just for genealogy. One thing it will help you understand your surname and its labels. How do you spell your surname? Well, if you look at the Y-DNA as reported by the Big Y, if we show you with this study, you will have a SNP name that could act as your surname because it stays the same and goes through time and it doesn't have misspellings. And then you can also see what other surnames are close to yours based on your genetics, your DNA. And with the advanced tools we're talking about, you'll be able to figure out when and where the splits were. And you can use both the STIRs and the SNPs, which we covered in the previous lectures, to define your family tree. So to repeat, it's useful for figuring out your name, your haplogroup, your haplotype, whatever word you want to use to describe that name. It's useful in figuring out surname studies if you're in one of those lines where surname has been consistent, and it's even better if you don't have a surname. It's good for finding cousins, and it's very good for testing whether you're related to somebody else or not. Now, to give you an idea of what's happening in the market, as well as the prices have come down, the amount of detail they can test has gone up. And the most popular branch of the male Y chromosome, which is the R branch, it's the one that there are more people than any other testing. You can see on this slide that back in the year 2002, there were 16 known branches. And as you come down through the time periods there, you see it went to 24, 71, 290, and in 2016 it was up to 10,000. And now we're getting branches that are unique for each person that was alive in about 200, 300 years ago. Here's an example that we'll be using from my work. My genealogy work of some 30 years tells me there's a person named Casper Rader who was alive in Virginia. He was born in 1732, and these are just 3 of his children and their descendants down to the day. And looking at the bottom part of that chart, you can see the blue arrow at the left is pointing to one of his descendants who's called Alton Rader. The one with the green background is Harry Rader. I'm the blue guy at the bottom, James Rader, and then Earl Rader's at the right. And it's one thing to notice there's a different number of generations between us and the same guy, because some families, you come from the firstborn, sometimes you come from the lastborn, sometimes you just come from a couple that gets married earlier. And here's the actual result from my Big Y test. And you'll see the column on the left has the name of the SNP, the thing that would replace your surname, so to speak, from genealogy. The green column are my results, places where I match. The location, in other words, that's a distance from the beginning of the Y chromosome. The column titled Harry, which does not have color, is another cousin. The blue column is a third cousin. And the fourth column, which is also not colored, is Alton. And these are the people we tested, and these are the results that we got, which will explain the new branches and so forth as we move forward. But the bottom line, let's show you what you get before we tell you, I'll try to motivate you to watch this. What do you get? This is a chart of the descendancy, the trees. And on the left, you see in gold first at the top, the company YFULL is the one that's doing the analysis. This is the version of their tree, which is 6.01, and they are the fastest in updating their trees and adding new data. It's one of the things that makes them good. Then you'll see also in gold, down about an inch, 2 inches from the top, you see R-207. That's a major group of people who were born about 3,000 years ago. And if you follow the lines underneath that, there's a sort of a branching. You see R-207 in green is under R-207. Then RCTS 43 is right below that. And then RY 15783 is below that. But off of RY 15783, there's sub-branches. And you'll see down at the bottom of the page, that arrow is pointing to the 4 people we talked about before that I tested. And this test shows that their common ancestor, based on DNA, was alive 275 years ago. In other words, these SNPs, these names, were valid names in the year 1800. So when we think about how far back that is, how did we get there? Well, if you look at the number of errors in the human between the beginning and now, and divide it out by time and number of SNPs, you'll find it varies between 110 and 170 years is when there's a new SNP. Okay, so now we have the average on the previous slide. On this slide, we're looking at a report from YFULL with 2 DNA tests back. 2 people of those 4 that I showed you on my descendants of Casper. And if you look with the red arrow, shows the average of these 2 is 2884. But if you look at the last 2 lines on the slide, you can see the bottom row says he has 22 SNPs, and that calculates out to 3399. The one above him only has 15, and that's 2370. So those are 2 of us that are fairly closely related by genealogy that have considerably difference on SNPs. What happens if we add another one? We're at 2884. We add another one. It brings that age down to 389. Now, why has it come to 389? The number of SNPs is they've got 2 that match real closely, and there's only 1 SNP difference between what they have difference. So that comes up to 389, and you can see on the far right on the bottom 3 rows, 216, 212, and 740. They average that to get to the 389. What happens if we add a 4th one? Now you notice here we're at still around 400. Let's add 1 more DNA test. Now we're below 300. And you notice again the calculation has changed again because the mix of the relationship between the 4 people. And you can see we have 2 of them that match exactly. And then 1 that's off by 1, another one's off by 4. So now we've got an average of 269. So we now know that according to this, our Casper Rader was born 269 years ago. Well, if we look at the calendar and the cemetery tombstone, we'll see he was born 285 years ago according to that calculation, and that estimate was 269. That's good enough for me if you ask me. That's right on. Did you figure out what was happening with those calculations? You notice that as you get more matching SNPs, those are considered as happening before your common ancestor. and the unmatching SNPs are counted after that. So at the last calculation, we had people that were 0, meaning they matched on everything and there was nothing further, so there was no further future. So they were at 0. And then there were a couple that had a few more SNPs, which averaged a small number, which brought us to about 275 years. So basically, when you get matching SNPs, it makes them considered as before your common person, which is logical. Now, as we start looking at things in more detail, those same 4 guys tested. On the left column, you'll see a highlight in yellow. I did an additional step. I went to the YFULL company and had them run a SNP test just on those 20 or so SNPs that my test showed, 23 SNPs. We couldn't do them all because there's actually 25 if I remember right, but there's some that were in the curlicue part that the Sanger method that YFULL uses can't test. But you notice the places where there's orange and red in the chart. Theoretically, these 4 people come from the same man, and any errors he had, they should all have, and any errors that they pick up beyond what one another has would have happened in the last little bit. But when you look at the difference between the 28 of Earl and the 16 of Harry, that means there's 12 SNPs difference. If you got 12 SNPs difference, and we're talking about over $100 years per SNP, that would mean they should be 1,200 years apart. So there's some variation in the quality of the test. And when I talked to the guys that run the test, I talked to them about Harry's, and I said, well, how come he's missing so many that the rest of us have? They said, well, would you like me to bump him up? Which means their test is working on a percentage. Family tree DNA has one way of looking at it, and if you look at YFULL, they have a different way of looking at it, and each one is how much they believe the test has to match before it's counted as a good match. Here's a little closer so you can see the numbers and the distances and the red and the green and the blue. But let's go on over to the chart. This chart is off of YFULL, and it's off of version 5 as opposed to 6, which is the new one today. But you look at the blue arrow on the left, and that's showing that Z207 we looked at a while ago that was over 3,000 years ago. We look at where it says FTDNA on the left. That's what Family Tree DNA's analysis says the most recent SNP is. But YFULL goes a little further and says, no, there's one more deeper. And you can see the time frame. They both show that both choices were done 29 years ago. That SNP occurred, but the last one with the red arrow next to it shows 400 years ago apart. So this science is not real tight science. It's a lot of estimations and statistics and mathematics. This is a report from YFULL, YTree version 6.01, and you'll see across the top it shows the SNPs that occur prior to where we are, starting with A0-T and going all the way across with some R's on the right, and then there's a second row where we have RZ206 and 208. And after 208 is 207. So that's what we're looking at in this picture, is that area of 207. And under 207, we've got 3 showing here that are sub-branches from 207. And under those, we have sub-branches and sub-branches. So when we look at mine, which is that group down there at the bottom, RFGC35930, where there are 9 people together with the U.S. flag on them, because they're all from a guy who was from Pennsylvania in the colonial era. And you can see from this chart who's related to who, when did it happen, and a whole bunch of other information. The more you study it, the more you work with it, the more you'll see about it. I see that there's some new people matching. And so I emailed them through the mail through YFULL. And this is what I told them. I said, I see that you, and I gave them the test number of the other person, are my closest matching group. Your group is 450 years ago, and mine is 275 years ago. The time of mine was 475 when I had just 2 tested, but when I added 2 more testers, it reduced to 275. My ancestor by genealogy was Caspar Rader, who arrived in Philadelphia in 1750. Birth unknown, place unknown. He retired to the Shenandoah Valley after 1800, where he died in 1812. I am Jim Rader, jim@rader.org, and I live in California, USA, and here's my phone number. So what is this all about? What are these sciences? How do you learn more about it? Well, if you really want to dig into it, there's 3 names you can look for publications and books by. Molecular anthropology is what the anthropologists call this. Genetic genealogist is what the geneticists call it. And population genetics is what the statistics people call it. And let's look at that a little more actual results. If you look at this group of people, up at the top, these are all type A. Not type A, what are they? I don't know. Anyway, there's a bunch of Greeks up there at the top, you see? And the Greek people are showing that they were 1950 years ago. And then in the same group, indented just a couple indentions, shows Ashkenazi Jews at 550 years before present time. So you can see here that it's suggesting that the ethnicity doesn't mean a whole lot, that you can go from Greek to Jewish people just by staying put. Here's a chart where they're trying to show the different cultures that appear in anthropology. And most of those words are not words I even know, let alone the names of countries, because this is way before countries. It's showing the Roman Empire down below the dotted line. Okay, let's try another subject in the same area. I thought back when National Geographic, who had their National Geographic project for DNA, would be the place to go to find out where and more geographic type things about the DNA. I've now gone to them 3 times, each time with about $200. The last time I went to them was with their new helix test, because it had all kinds of suggestions. They were measuring all kinds of other things. And while the test was being worked on, I got a hold of the guy that runs the project at National Geographic, and I gave him the details of my DNA results from other companies and how they were showing much, much more information than National Geographic was. And I had hopes that since they upgraded their tests, even with a new company to test with and all that, they would provide more information. But basically, this chart of where I came from and when is unchanged to a large degree, the top part especially, and most of it was incomplete. As you'll see, there's a bunch of later type stuff. And in spite of the fact they've had 12, 14 years to work on it, and they've been paid $200 3 times. But here's what their results added to, with a few other things trying to add to this from other companies, say my ancestral Y-DNA was found. Remember that those SNPs, as we have them through time, each one happened at a particular time in the history. So that we're going to start at a period of over 100,000 years ago in Africa, which is where branch P305 is. As we move forward in the time, you'll see we go 80, 70, 60, 55, and 50,000 years ago with these list of SNPs down to M578. And we've moved from Africa to East Africa to Southwest Asia. Now, as we move forward, we go to 45,000 years ago, 42,000, 35,000, and we finally get to where the common European type DNA comes, Y-DNA, which is called R, which is composed of R1A and R1B and a bunch of other things. But the SNPs that define that are like M207, and that's age 30,000 years ago. And you see we're bouncing around Asia. All of these on this page are basically saying something about Asia. and we got down to the bottom 343, which has got a broader range. It says 17 to 22,000 years ago, and we're still in Southwest Asia. Then we go to the next page, and they generally say Asia, but they have no information on those top three. The bottom one, M269, is a very common one, and you'll see we've got a more specific naming of the old naming convention on this chart too, to let you know where you are. We're in R1, B1, A1, A2 at this level, and that's 8,000 years ago. So that might give you a hint that if you only get that few numbers, or you get something around M269, we aren't getting the genealogy time frame yet. We're 6,000 to 15,000 years ago, and we don't know where, someplace West Asia maybe. Then we go to the next group of SNPs that are occurring. We work down through 8,000 down to 6,600 years ago to P312, and you notice that R1B1A1 has some more letters after it. That shows 16% of the population of France has this type of DNA between 5,000 and 14,000 years ago. And we can move forward now in time, and we're going to go down to the SRY2627 with a big green box around it. At that point, I've got about 14 letters after my R1, and it gets so cumbersome and you can't work with it. So we start changing what we call it. Instead of saying R1 and all those letters, we just say R-SRY2627. And then we go down with two more on the page with R-Z202, Z205, and we're 4,300 years ago at this point. Then we go to 4,000 years ago with more letters in Z207 and 208. And remember those charts we were showing you, the results started with 207 at the top. And we quickly came from the 4,000 years ago down into less than 1,000 years ago. And this was on YFULL's tree 410. And you can see some of my SNPs at the bottom, one of my new SNPs that are highlighted in green.